So my talk today will be a bit unusual for me in that the subject differs a bit from my usual research. In a normal day, I'm mostly concerned with two research avenues. One is the systematics and biodiversity discovery of parasitoid wasps of the family Ichneumonidae. So much of my work is devoted to building large-scale phylogenies and revisionary systematics, including the description of new species. And the other one is the study of phenotypic evolution and how it relates to various ecological traits as a result of convergent evolution. But today I want to talk to you about a third, more recently developing aspect of my research, which is the use of whole genomes to understand biological adaptations. Specifically, I want to talk to you about what I think is one of the coolest biological systems in the planet. That is, the domestication of viruses by parasitic wasps into endogenous genomic elements that they use as a weapon against their hosts. The recent popularization of whole genome sequencing techniques has enabled us to gain a lot of insight on the genomic architecture of these interactions and how they have evolved across different wasp lineages. So that's what I want to talk a little bit about today. All of the work that you see here today has been done in close collaboration with partners at the Smithsonian Institution in the US, especially my former postdoc supervisor, Sean Brady, and bioinformatics specialist, Rebecca Dickel, and also with researchers at the INHI, virologist and Nathalie Volkov, and genome biologist Fabrice Legueay and Stephanie Hobbin. They know way more about viruses and genomes than I do, and they have really been the people really driving the technical elements of the hardcore genomic work of this project. I think there's someone with their microphone. Okay, I think, unless it's me. Is this my microphone? Well, can you hear me?
Yes, we hear you.
Oh, okay, thank you. Okay, it's just I was hearing some noise and I was not sure if it was me or elsewhere. Thank you. So moving on to the topic of the talk, we will all the time be talking about parasitoid wasps, so I think it's important to do a short introduction. These are insects that are free living as adults, but they develop as parasites in their immature stages. So the female laid their eggs into other arthropods, and the wasp larva then gradually consumes its host, eventually killing it. Some parasitoids, they can permanently paralyze their hosts and simply eat it slowly as if it were a giant bag of meat. But there are other parasitoids that they let the host keep active and feeding after being parasitized. So these are called coinobiont parasitoids. So for instance, the reason why this wasp here on the left can lay its egg in such a small caterpillar is that the caterpillar will keep eating and growing while the wasp larva slowly consumes it from the inside. As you may imagine, the host doesn't really like to be slowly eaten alive, and so these insects have several defense mechanisms against parasitism. Just like we have white blood cells that attack foreign structures inside of our body, the insects also have specialized defense cells that can congregate around a parasitoid egg and destroy it. It's a process called encapsulation. And the parasitoid, in turn, needs to then develop strategies to avoid this to happen, to avoid its egg to be encapsulated. And perhaps the most impressive of these strategies is the use of symbiotic viruses called poly-DNA viruses or polytnaviruses. These are viruses that are injected by the parasitic wasp into the host along with the eggs. Once inside the host, they express genes that will help to disable the host's immune system, which would otherwise encapsulate and destroy the egg. This is pretty much the only known case of obligate symbiosis between a virus and an eukaryotic organism. And on top of that, polytnaviruses are also quite unusual viruses in the genomic level. Their genomes are large, well, large for a virus at least, and they're divided into several separate circular fragments that is called, they're called segments. It's much like human chromosomes. The genome is divided into separate different molecules of DNA, which is pretty unique for viruses. And that's the origin of their name, poly-DNA viruses, because there's several different viral, different DNA molecules. Now, the catch here is that the viruses, they don't really reproduce inside of the host. They only express genes. So the viral particles, they're only actually produced in the ovaries of the adult wasp. And the whole viral genome is actually integrated into the wasp genome. So the way that new viral particles are produced, instead of what you have regular with the infection and reproduction inside of a host cell, is that they are clipped out of the wasp genome and packaged so that they can be inoculated into the host. So this represents a totally different life cycle than what we're used for viruses, right? So what we usually see is this here on the right. A virus infects a host, where it generates new viruses, which in turn infect a new host, and so on and so forth. But instead, what we have here is we have a wasp that produces a virus that infects a host, which enable the wasp to produce a new wasp, which in turn produces new viruses, etc., etc. I mean, at least to me, this life cycle is kind of mind-blowing. It's totally different than what we see for viruses, for wasps, or anything else in the planet, as far as I know. And the most surprising thing is that this extremely complex and specific life history trait has actually evolved more than once. So Polytna viruses are found in two parasitoid wasp families, the Braconidae and the Ichneumonidae. In both groups, they're restricted to only a subset of the species in the family, so we know that they have evolved separately rather than being inherited from the ancestor of both families, both families, yeah. Polytna viruses found in Braconidae are called brachoviruses, and they're found in a group of subfamilies called the microgastroid complex, which includes tens of thousands of species. Polytna viruses found in ichneumonids, on the other hand, are called ichnoviruses. I know the nomenclature is not the most creative, but it's what we have. And they're found in two subfamilies, the Bankini and the Campoplegini. I think you'll agree with me that this whole thing of parasitoids incorporating viruses into their genome and then using them as biological weapons is a very intriguing phenomenon. And it leads to a lot of questions about the evolutionary aspects of this interaction. For example, how did this viral integration occur in the first place, and how many times? Which groups of ancestral viruses were recruited by these wasps? How is the viral DNA distributed in the wasp genome? What genetic features are conserved or diverge? Now, we actually know a lot about brachoviruses, the viruses found in braconid wasps. Researchers have sequenced the genome of braconid wasps and mapped the insertions of viral segments in there. And they found that they are, And they found that they are present in the wasp genome in a few large clusters. So you have a big part of the wasp genome, and you find these segments one after the other in a chain. And these viral segments are also bordered by excision motifs that signal the site of extraction and packaging of the viral material. So there's a lot of knowledge accumulated of how the virus is organized into the genome of braconid wasps. Another big discovery was defining that the genes that control the replication and the structure of the viruses, they're found in the wasp genome, but they are not packaged into the final viral particle, right? If you remember, the virus, it doesn't replicate inside of the host. So these genes are not actually packaged in the final virus because you won't need them. So instead, they are only found into the wasp genome. And by analyzing those genes, Anne Bezier and colleagues noticed that they are highly similar to core genes from a group of genes called the nudiviruses. And then they concluded that brachoviruses probably descend from an ancestral nudivirus that got incorporated into braconid wasps. In the meantime, a separate study on the phylogeny of and divergence times of the wasps that brachoviruses have found that these association evolved over 100 million years ago. So it's really beautiful to see how these different lines of evidence complement each other to build up the knowledge of the structure and the evolution of brachoviruses. On the other hand, until very recently, we knew relatively little about ichnoviruses, the viruses found in ichneumoniid wasps. partially because no one had ever sequenced the genome of an ichneumoniid carrying polytenoviruses. One important discovery that has been made is that ichnoviruses, they have that viral machinery that I just mentioned that has the structural and replication genes. They have a similar viral machinery that doesn't get packaged into the viral particle. These are called the ISYEBs, or structural protein encoding regions. And they have those, but on the other hand, they are not similar to any known group of viruses. So the analysis of those regions do not allow us to identify from which viruses ichnoviruses came from. So maybe they come from an extinct lineage of viruses. We don't really know. There's more mystery than answers at this stage. So to start to understand the genomic architecture of ichnoviruses, we sequence these collaborators from the INHI, from the Smithsonian, and myself. We sequenced the genome of two species from the subfamily Campoplegidae. What we set out to find out was, first, how are these viral insertions distributed in the ichneumoniid genome? Second, is the genome architecture across different species conserved? And third, is there a difference between proviral genes and the viral machinery? That is there a difference in the way that in the genomic organization of the proviral genes, that is the virus, the genes that get packaged and delivered into the host, versus the viral machinery that makes up the structure and replication of the virus, and that doesn't get packaged into the final virus particle. So we sequenced these two different Campoplegidae species. We used only it's Campylidis sonorensis and Hypozoeter didymater, and we used only Illumina sequencing, so only short read sequencing, but at very deep coverage. And for Hypozoeter didymater, we also used mate pair libraries that increase, try to get better assemblies with these mate pair libraries. So in the end, we were able to get assemblies of quite good quality, even though we only used short read sequencing. So we got scaffold sizes up to 15 million base pairs, which is pretty good. If you look at the overall statistics, including estimated genome size, N50 of the scaffolds, and so on, everything was on par with other projects of sequencing whole genomes in parasitoids. So we got very similar, or within range statistics for all the assembly metrics. And to assess if these genomes are really, really complete, we also searched for a set of BUSCOs, which are single copy ortholog genes that are used, among other things, as a reference to check genome completeness. So we tested our assemblies against a set of BUSCOs that are universal for all the insects, and we found over 99% completeness. Again, with similar statistics for BUSCO completion as found in other genome sequencing projects in Hymenoptera. So we're pretty confident that these genome assemblies, even if they are not at the chromosomal level, they contained all of the genes that are found in the genome of these wasps. So we then looked for polytenovirus elements in these two wasp genomes. Remember, here we are looking for two kinds of functional components. The first one are the proviral segments, which correspond to the virulence genes that are injected by the wasp into the host. The second one, the ISYEBs, correspond to the virus-making machinery that controls the replication and the particle production, and they're not packaged into the virus particle. They only exist in the wasp genome. Now, at this point, you might be wondering, how exactly are we going to look for these viral elements in the wasp genome? Do we even know what we're looking for? And well, yes. So for both of these species, the proviral segments had already been sequenced directly from the purified virus. So we searched specifically for these sequences in the wasp genome. For the ISYEBs, we knew the sequences for one of the species, and so for the other one, we looked for those genes on the basis of similarity search compared to the known ISYEB genes. So okay, this is all pretty exciting. Will we find anything in the wasp genome when we look for these viral elements? And the answer is yes, we found them. So let me show you first the distribution of the proviral segments in the wasp genome. Here, each of these black lines represent a scaffold, a piece of the genome. And the horizontal red bars represent the viral segments. And we can see that they're not in clusters or grouped at all. In fact, they seem to be spread out pretty much everywhere in the wasp genome, separated one from each other by large chunks, large pieces of wasp DNA. So this is interesting. The proviral segments, they're spread out everywhere, and they're also in different regions of the genome. The areas where you find the areas where you find each segment in the two wasps are different. So if you look at the flanking regions, the regions that flank each viral segment or each viral gene in the two wasps, they're different, indicating that these proviral segments are able to move in the genome of wasps, or that their location is not conserved. Same thing with the gene order, and in many cases the gene content, they vary a lot, indicating that there's a lot of variation in the proviral segments from one species of wasp to the other. We confirmed the spread out nature of these proviral segments by using a FISH assay, a fluorescent in situ hybridization, that used RNA probes matching 4 of the viral segments. And then it turns out that each one of these 4 segments that were tested is located in a different chromosome of the wasp. Now, on the other hand, the ISYEB, that is the virus-making machinery in the two wasps, seems to be very, very conserved. Remember, these are the genes that are related to viral replication and to the structure and formation of the viral envelope, but they're not included into the final virus particle, so they do not get delivered to the host. As you can see here in the mapping of these replication genes in the genomes of the two wasps, we find that these are clustered in just a few places. They are largely in the same regions of the wasp genome, and they also share most of their genes. And these genes are largely in the same order. So these bars are kind of pointing matching the corresponding genes to each other. So you can see that they are largely in the same order and in the same areas of the wasp genome between the two different species. So if you think of the functional roles of these two components, this kind of makes evolutionary sense, right? The virulence genes, the proviral segments, they need to be fine-tuned to the host physiology, because they are the molecules that directly interact with the host physiology and the host immune system. So it makes sense that they need to be rapidly evolving across different species. Meanwhile, the structure of the replication machinery was probably inherited from the ancestral virus and did not have to change much, because it's still doing the same thing that it was doing originally, which is to replicate the virus and build its structure. So it did not have to change much over time. So these results were published some time ago at BMC Biology. In this paper, we also investigated other things, such as how the virus gets clipped off of the wasp genome, differences between brachoviruses and ichnoviruses, etc. So if you want to get more in-depth information about these topics, there is your reference. So this paper is very much about the hardcore genetic aspects of the genomic architecture of the polyadenoviruses. But as an evolutionary biologist who thinks more in comparative terms, the thing that interests me particularly the most is the phylogenetic distribution of the polyadenoviruses across the wasps. So remember, we have discussed that these are found in 2 different families. The polyadenoviruses are found in 2 different families, the Braconidae and the Ichneumonidae. And within the Ichneumonidae, they are found in 2 different subfamilies, the Benchinae and the Campopleginae. But the existing phylogenetic information shows that these 2 subfamilies, they're not sister groups. That is to say, there are lots of other lineages in between the 2 of them, right? So there has been some effort to screen these other subfamilies to see if they have polyadenoviruses, both by direct dissection of the ovaries and also by screening their genomes with low coverage genome sequencing. But these efforts have been pretty sparse, but so far we haven't really found, I mean, I'd say the studies existing haven't really found any evidence of other viral elements in these other subfamilies, right? So In fact, even within each subfamily, the viruses are known only from a handful of species. So the sampling, the screening of different species to see if they harbor viruses or not has not been very thorough, very extensive in the family as a whole. So the current consensus is that this viral association evolved actually independently twice in the Ichneumonidae, once in Banchine and once in Campoplegine. But also, there's this big and largely untested assumption that is usually included in the papers about polytenoviruses, that is that all members of each subfamily have these viruses. And we don't know that for sure because we haven't really screened a lot of species within each family, within each subfamily for viruses. In fact, we can already see that the situation is a bit more complex than that when we consider the case of the species Venturiae canescens. This is a species of Campoplegine, one of the subfamilies known to harbor polytenoviruses. But instead of polytenoviruses, this species has what is called virus-like particles. It's a molecule that resembles a virus, but has no genetic material. So VLPs in this species play a role similar to polytenoviruses in delivering virulence proteins to the host, but they're very clearly not poly-DNA viruses because they have no DNA. In 2015, the super important study by Pichon and collaborators located the viruses, the genes responsible for the production of VLPs in the genome of Venturiae canescens, and found that they closely resemble nudiviruses, which is the same group of viruses that gave origin to brachoviruses. And the same study also found that the genome of Venturiae canescens contained contain remnants of polytenoviruses, remnants of the ichnoviruses, in the form of genes called repeat element genes, or rep genes. This is the largest gene family that is found in polytenoviruses genomes, but they still have unknown function, right? But they are, there's lots of them inside of the polytenovirus genome. So the conclusion of this study, of course, was that Venturiae canescens must have undergone a sort of virus substitution process. The loss of the capacity to produce viable ichnoviruses, along with the domestication of a nudivirus that played a similar role and that replaced the ichnoviruses, either because it was more efficient or, for reasons still unknown. But that was the idea that emerged from that study. So that certainly raises a lot of additional questions, right? Did this happen only in this species, Venturiae canescens? How easily then can these viral elements be lost or gained if we have such a clear example of a new viral element happening in this species? So clearly there's a lot of interesting things to discover when we start to look comparatively at a larger assemblage of species of parasitoids. So in order to start looking comparatively at viral elements, we need two things. One are multiple whole genomes, of course, and the other is the reliable phylogenetic framework. That is, how do these species relate to one another? So luckily, as I have mentioned in the beginning of the talk, my work on a normal day is actually exactly to generate these large-scale phylogenies for ichneumoniid wasps. So at the present, most of my work relies on genomic ultra-conserved elements, or UCEs. As the name says, UCEs are small genomic regions that are highly conserved across all species from a given lineage. So once we discover these UCEs in existing genomic resources, we can, for example, screen the known genomes of Hymenoptera for UCEs and identify these highly conserved regions. We can design specific RNA probes that correspond to these regions. So, and then instead of the regular PCR Sanger sequencing approach, we can hybridize these RNA probes with our target DNA and then pull out those fragments out of the main DNA extraction and sequence everything at once using next-gen sequencing. So the UCE region itself being ultra-conserved, it's obviously not very informative. But as you go towards the flanking regions of the core UCE area, there's a lot more variation that can then be used for phylogenetic analysis. Right now, I'm using an existing probe set that targets over 2,500 loci, that are conserved for all of the Hymenoptera, that is all wasps, ants, and bees. Another great thing about UCEs is that because they're mostly small fragments of DNA, they allow us to generate data from highly degraded DNA. So really old museum specimens, sequence material from specimens of over 70 years of age, which would otherwise not be available for phylogenetic work. So this is work that has been done with several collaborators from around the world for the past few years. Our data set currently includes over 800 species of Ichneumonidae from all its subfamilies and represents the first really robust phylogeny for the family as a whole. And this allows us to examine any results based on whole genome sequencing in a sort of evolutionary comparative framework. These results, for example, confirm previous studies that show that Benchini and Campobleggini are not sister groups, but it also provides information about the internal relationships of the species in each of these families, which is obviously very useful for us to understand, for any effort to understand the evolution of viral association in these subfamilies. Meanwhile, we've been also working hard on the generation of whole genome sequencing data for more and more species of Ichneumonidae. This is ongoing work, but so far we've been able to generate short read sequencing data for 96 species of Ichneumonidae sampled across the family, mostly at between 20 to 60x coverage, as well as also a little bit of long read data in the form of Oxford nanopore data. So nanopore is sort of an emerging technology that allows the generation of very long reads up to 1 million base pair reads and so on. But regularly you can get N50s of up to 10,000 base pairs. So these really long reads, these really long DNA fragments that you can sequence, they are obviously very useful for building larger and more complete assemblies. So even if you have relatively less data with nanopore than you'd have with Illumina, you can often dramatically improve your assemblies by doing this. And it's also a pretty interesting technology because the sequencer can be just this tiny machine the size of a large pen flash drive, a USB card, and you can do everything in your own lab and so on. But we can also talk about this in the question section. It's pretty interesting. I got this machine in March 2020, and then a week later the museum closed due to COVID, and it was 3 months closed. And then I had to figure out by myself how to use the machine and all the protocols while only being able to go to the museum once a week, with everything closed. It was quite the adventure. But we haven't been able to do in-depth analysis of the whole data set. Oops, there's something wrong here, but I think we can make do. So we haven't been able to analyze the whole data set yet, but for the seminar today, I want to focus the discussion specifically in the 2 subfamilies that we know that harbor viral symbionts. So we set aside short genome assemblies for short read genome assemblies for 24 species. So there's 14 Campoblegginae, 7 Benchini, and 3 outgroup taxa. These assemblies varied a lot in quality, with N50s varying all the way from 1621 base pairs, which is very low, to over 74,000 base pairs. The longest contig we assembled for any of the assemblies were 673,000 base pairs. So this is a sharp contrast. You notice this is a sharp contrast with the assemblies for the other 2 species that we have that were, super complete with scaffolds of millions of base pairs. So I'm happy to discuss the sampling, sequencing, and the assembly methods and issues that we had in more detail during the question time. But immediately these raises concerns, right? Can we actually learn anything from such low coverage and fragmented assemblies? So again, the first clue that we can have is go look at the Busco completeness, right? So these are the universal single copy ortholons. So we ran these analyses to test if these highly fragmented assemblies still had a lot of complete genes in them. And actually, they did. The fact that even with these fragmented assemblies, the fact is that even with this highly fragmented assemblies, we could still find an average 90% completeness for the buscos loci, except for one outlier, which was a species of Duzona that had really, really bad assemblies. But if you take the set of all of the other species together, we got an average of 90% busco completeness, and in some cases up to 99%. So, this is encouraging. Even maybe these assemblies will be useful for something after all, even if they're highly, highly fragmented. So at this point, of course, I cried for help to my colleagues at INHI, and they kindly took over the bioinformatic analysis for this step. Again, this is still ongoing work, and there's still a lot left to do. But so far, we've blasted open reading frames for our new assemblies against proviral and proteins from the proviral segments and the EVE spurs found in the complete genome of Hypozoeter didymater, and also against the nudiviral genes found in Venturiae canescens. Our expectation, or our null hypothesis, so to speak, was that all of the Benchini and Campoplegini would have polydnaviruses, and that none of the outgroups would, because this is what is sort of tacitly assumed in the scientific literature. What we found, however, it's a bit different. So here's a phylogenetic tree of the examined genomes, and on the right column, we signal the presence of either EVE spurs, indicating the presence of polydnaviruses, or nudiviral genes. So first, in Benchini, most species did have EVE spurs, as we expected, but there was one that didn't have them. So this was Benchus vitosus, which is sister group to all of the other genomes, and all of the other ones did have EVE spurs, which indicate the presence of polydnaviruses. In Campoplegini, the situation was even more unexpected, perhaps. We found that the subfamily can be divided into two large clades, and that EVE spurs were only found in one of them. So the other clade had several species that don't seem to have any endogenous viral elements, but in 3 species we found clear homologs to the nudiviral genes found in Venturiae canescens. So it seems that it's not only Venturiae canescens that has that association with nudiviruses, but other species as well. So we have 2 clades, group A, I'm calling them here group A and group B. So group A has no indication of polydnaviruses, but a subset of this group that I'm calling group A1 does have nudiviral genes. And group B comprises the species that have the EVE spurs, the viral machinery that indicates the presence of polydnaviruses. Now, what we can do as well is to get these species for which we have whole genomes and then plug them into the big Ichneumonidae phylogeny that I've generated with my UCEs. So what we can do here is that we can pull out the sequences of ultra-conserved elements in silico bioinformatically, and then add those genomes, add those species for which we have the genomes into the phylogenetic matrix. So here, so for example, we can do this here in Benchini. So here are in bright blue the species for which we have genomes, and in white are the species that were only sequenced for phylogenetic purposes. So this helps us to put the evolution of ichnoviruses in the group in context. So for example, it indicates that polydnaviruses are probably absent in the tribe Benchini as a whole, which is the group, the clade that includes Benchus vitosus. And it can also help us to point out more or less the exact point where the association with polydnaviruses appeared in the group. So the phylogeny here offers 3 possible branches for where the domestication of viruses would have occurred, that it's in between the point where we find no viruses and the point where we find viruses for all of the members that we sampled. And so this also helps us to suggest further species to sequence in order to find the exact point of transition between lineages without viruses and with viruses. So for now, for example, I would really like to put my hands into some samples of Pantelis and Sacklebenia, because those would be very interesting to sequence for whole genome sequencing and see if they also have indication of viral elements in them. Likewise, in Campoplegine, we can also trace the viral associations along the phylogeny. And clearly, we can see that two different lineages in the subfamily independently evolved viral associations. One clade has the ichnoviruses, the already well-characterized ichnoviruses, as indicated by the presence of ISYEB. And the other clade, on the other hand, did not lose the ichnovirus as previously assumed, but they seem to have never had them. They seem to have had no viral elements until they acquired a new virus and domesticated it through an analogous process, but a distinct process, right? Because it turned it into virus-like particles instead of polypnoviruses. So we have these two analogous processes independently happening in the subfamily Campoplegine. And likewise, here we have three possible points in the phylogeny where we could point out that's where polypnoviruses occurred. And again, we have a few more genomes to sequence in order to refine our understanding exactly which lineages harbor polypnoviruses. Finally, there's one more big surprise revealed by our analysis. So the family of proviral genes, the genes that are found in the packaged viral genome, there's two families of genes in those in the packaged genome of the virus. They're called vancurins and vinexins, and these they encode virulence proteins that help to prevent the immune reaction of the immune response of the host. So these are virulence genes. They're delivered by the wasp inside the virus, right? But what we found is that they were not exclusive of the wasps that had polypnoviruses, but instead they were found in every single species that we screened. And the repeat element genes that make up the largest gene family in the genome of polypnoviruses were also found in species that harbor polypnoviruses. So, these are indicated here in dashed lines. These are the groups that have polypnoviruses, as indicated by the presence of ISYEB. That is the virus-making machinery. So you can see that all of these species actually have vancurins and vinexins, and that many species that do not have polypnoviruses also have the repeat element genes. And we have to consider as well that for the zona in which these repeat elements were not found, that these were the poorest, the two worst assemblies with the lowest Busco completeness. So it's possible that those genes may be there, but were just not found due to the incomplete nature of these assemblies. So this is interesting because it provides strong indication that at least part of the genetic material that is encoded inside of polypnoviruses actually has wasp origin. And this is something that had been suspected before, but it's very nice to see this confirmed in our analysis in this comparative framework, that you actually find these genes in the genomes of many other wasps. So I'm curious to add more outgroups, that is more wasps that have no polypnoviruses, to see how extensive this is. But essentially, Odontocolon, the two outgroups that we have here, Odontocolon and Rissa, they belong to very different lineages of Ichneumonidae. So it might be that vancurins and vinexins, at least, are a component that is present in the genomes of all Ichneumonids. So this is very nice to see. And so, in conclusion, what we can tell is that by sequencing whole genomes of wasps, by doing these more recently available techniques of genome sequencing that allow us to do several genomes in more or less inexpensive fashion, we were able to conclude several things in the past couple of years about ichnoviruses. First, we could map polypnovirus insertions in the whole genome of two species of Ichneumonids. We found that proviral segments and ISYEB, which is the viral machinery, have very different genomic architecture. We confirmed that at least some proviral proteins that are delivered by the viruses, they have wasp origin. We found that PDVs, polypnoviruses, are restricted to only a subset of Bunchini and a subset of Campoplegine. And we concluded that there's there have been two separate de novo, like new viral acquisitions in Campoplegene, and also that virus-like proteins derived from that are derived from a nudibvirus are not only present in Venturia canescens, which is the only species from which we knew it so far, but are probably present in a large clade comprising lots and lots of species of Campoplegians. So, in terms of future challenges that posed by by this line of research, one thing we have to point out, of course, is that there's an important limitation in all of these techniques of genome screening, which is that we're only looking for what we already know. So when we sequence the genome of a wasp and we look for viral elements, we're looking for sequences that resemble viral sequences that are already characterized and known. So there may be new things, new viral elements, new genes that we are not finding because they simply have not been characterized yet. And so, for example, there has been an important work by Galen Burke and collaborators recently that looked not only for known polytenoviruses and so on across ichneumonids and braconids, but also for any indication. They got a bunch of genes that are common across all families of viruses that are associated with insects, and looked for any indication of viral presence in the wasp genome. So that's also another avenue that can be pursued in the future with a large assemblage of ichneumonic genomes. It's also interesting to see, you see large parts of the ichneumonic tree where there seems to be no viral association, and then in Campoplegene we have two separate events of viral association. So from an evolutionary point of view, an evolutionary and ecological point of view, I'm curious to understand whether this subfamily is particularly suitable for viral domestication for any reason. What makes the Campoplegene special that two different groups acquired independently the association with viruses? And of course, the immediate next step is to sequence more Campoplegene genomes to be able to refine our understanding of the evolution in this group, especially the sort of polytenoviruses versus nudiviral VLP. So I actually submitted an ATM project proposal to the museum to see if we can fund that. So if you by any chance is asked to review this proposal, I hope that I convinced you today that this is an interesting subject. And with that, that's it. I'd like to thank you, and I'll welcome any questions if I can answer them. Thank you very much.
Thank you very much, Bernardo. Are there any questions?
Yeah, I do have a question.
Marwan?
Thank you very much. This was really interesting. I have questions about, I have two questions. First, did you do a decontamination of your genome, like before or after assembly, or just assemble straight with your reads and had like a 99% boost score?
We did that for the first study with the two species that we sequenced the whole genomes for. For the new assemblies, we haven't done it yet because it's still kind of ongoing starting work, but that's definitely in the plan. But for the two genomes that are already published, yes, we did.
Okay, and how did you do it? Which tools did you use to decontaminate?
That I won't remember, because that part who did it was Rebecca Dickel in the Smithsonian for Campolitis sonorensis, and the INHI team for Hypozoder. So I was mostly looking at the other evolutionary aspects and not the hardcore bioinformatics stuff.
Okay, thank you. And my second question is, you screened like gene families that you were looking for, and which program did you use to do this? Did you just do it manually, like providing a FASTA file with homologous sequences from known database, and you found your viruses genes in the genome? So you had like another way to do it more automatically, more extensively?
No, it was mostly manual. Yes.
Okay. There's no secret.
Okay. So far, yeah.
And for the Busco database you used, was it Arthropoda? I didn't hear it when you said.
Insecta. It was Insecta.
Okay. Thank you very much. It was really interesting.
Thank you.
Violaine?
Yes. Can you hear me? Yes.
Mm-hmm.
Thank you, Bernardo. Thanks a lot for your talk. It was really great. Finally, I understand a bit about these brachoviruses that I heard a million times about, but I never understand anything. So suddenly it's become very, very much clearer. My question was about the potential losses of those virus in some wasp species. So in some of the clades you show, there is some species that do have remaining viral genes and some species where you can't find them. So probably they are lost in those species. And I was wondering what is the selection promoting that kind of loss. And I was wondering whether host resistance would be an hypothesis. And could you, for instance, test whether there is a correlation between the loss of these viral genes and the host range or something like this? Did you already try to look at that?
So, just to check if I understood what you mean, you were asking about the potential losses of the virus. So
Yes.
So far, that's what I found interesting about our results. When it was discovered that the species Venturi canescens had virus-like particles, they also screened the genome and found remnants of the repeat element genes, which are genes that are found in the polytnaviruses. So they concluded, well, then this species must once have the virus and then lost it. But our analysis suggests that the repeat element genes perhaps have wasp origin. So they're actually originally wasp genes that got packaged and transferred. So the virus is actually used as a delivery mechanism for these proteins. But the proteins, the virulence proteins that are used to suppress the immune system of the host are actually from wasp origin. So what this indicates to me is that that wasp Venturi canescens that has the virus-like particle, it didn't lose the virus, but instead it never had it. So you acquired a new virus, which was the nudibvirus. And so far, we only knew it for that particular species, Venturi canescens. So our studies indicate that it's actually present in a large clade. So it was not a one-off thing, but an event that was spread out in a large lineage.
Okay, so that means that you almost never actually observe a loss of those viruses.
So far, no.
Once those viruses are settled, let's say, within a species, they are very unlikely to get lost.
Yeah, I think so. So in braconids, this is much better characterized. So there have been many more species that have been screened for the virus, and it seems, and for within this clade where they first evolved, which is the microgastroid complex, they never found any species that doesn't have them. So it seems that all of the species have them, because I think it's such an integral part of the interaction with the physiology of the host that if the wasp lost it, it would be hard. But one thing that is very interesting is that if you look at both the microgastroids, which are the braconids that attack, that are the braconids that have viruses, and the lineages of ichneumonids that have viruses, all of them are essentially parasitoids of caterpillars. They attack caterpillars. But in 2019, the team from INHI, Natalie Volkov, Stephanie Hobbing, and so on, they found a group of Campopleginae, a genus of Campopleginae that attacks beetles, and they also have polytnaviruses. So I'm super curious to sequence the genome of that wasp as well, to see if the genomic architecture changes or something like that, because obviously one thing is attacking a caterpillar and other thing is attacking a beetle. Yeah. So I'm super interested to find more about that.
Okay, cool. Come back to us whenever you will have the results. I'm very curious. Thank you. It was really a great, amazing talk.
Thank you.
Bertrand?
Yes, do you hear me?
Yes.
Okay, thank you. So, oh yes, I agree. It's a great talk. It's always a great talk about parasitoid. It's a fantastic topic. I have two questions that are linked. The first one is that, do you know the mechanisms of immune escape introduced by the virus, or more interesting, the VLPs? And the second question is, do you know if the genes or VLPs are associated with other functions, like, for example, behavior manipulation of the host, like we know in Dinocampus, for example?
Mm-hmm. So I'll answer the second one because I think it's easier. As far as I know, no, there is no behavior manipulation or anything like that. But it's mostly physiological changes that are induced. For example, besides the question of the immune system, I think also the development, molting, and so on are also impacted. So these physiological changes, yes, but not anything related to behavior, as far as I know. Now, for the first question, what is the mechanism for which the immune escape is promoted? So that is known. I am far from a specialist on the physiological elements, but as far as I understand it, what these proteins do is that they interrupt the chain of reactions, of cellular reactions, that lead to the formation of the hemocytes that attack the egg. So they can contribute to the apoptosis of the hemocytes, the defense cells, and they can also prevent them to form melanin and interrupt the chain that forms melanin and other elements that attack the wasp egg. So what they did essentially is disrupt these cellular reactions, but there is plenty of literature about the specific cellular mechanisms by which it occurs, but it's just not really my field of specialty.
Okay.
Violaine, do you have another question or?
Non, désolé, j'ai oublié de baisser la main. Pardon.
Oui. Est-ce que quelqu'un d'autre a une question?
Les questions en français sont bien aussi.
Otherwise, I would have one.
Oui.
Do you know about mobile elements that would be present in the different genomes and whether there's an interplay maybe in Campopleginae for domestication or the absence of in some families and viral and mobile elements interplay? So
in the previous paper, the BMC biology paper, we did try for that. I say we, but it was Fabrice and Stephanie, of course. We looked for they looked for the presence of transposable elements near the viral segments because we thought that this maybe would be an explanation of how they change so much. And what we found was that there was a slight indication of higher presence of transposable elements near the proviral segments. But when you corrected for multiple comparisons, this effect disappeared. It was not significant anymore. So, correcting for multiple comparisons is always a kind of like a tricky thing. Sometimes it's appropriate, sometimes not that much. So, I think there's definitely something to look for in there, but so far we don't have any strong evidence that is the case.
Thank you very much. Other questions? Well, then thank you very, very much. It was really very interesting.
Merci beaucoup.
Merci.
Merci
au revoir à tous.
À tout le monde, oui, au revoir. À la prochaine.
Au revoir Bernardo et tout le monde. À bientôt.